So I finished processing the resting state data. Now I'm going to show you looking at what we call second level results. So these are really, for the most part, the results that we're interested in with functional imaging. And so what I'm going to do for here is load up a MATLAB session on HyperGator. And I have this set up from what I had the other day. 2 cores, 16 gigabytes of RAM, 1 core should be fine, class drive. I'm going to request this for 24 hours. Don't need that long. And we'll click launch. Now this will start at some point. Again, it depends on how busy HyperGator is, so it could take anywhere from seconds to hours or longer to start. But as we saw, that was quick, so I can launch MATLAB. And remember, you need to have SPM and CONN added to your path. That has been done already, hopefully, in a previous video. One way to check it is to go to set path and to see, do you have the CONN and SPM in there. If you don't have that, go watch the other video. And I'm going to cancel saving the path because I really didn't make any changes to it. So the first thing I'm going to do is change to the location I need. So we can use that CD command, and then here is where I have the directory. This class resting state fMRI is where I have a file. But I'm going to come here, I'll type in CONN, all lowercase, and that will load the software. And then I resize the window, and I'm going to let this load. It doesn't take very long, but I will come back when it's loaded. Yeah, this is loaded. It really didn't take that long. I could have waited. But I also went over to the Files app, and I loaded in my directory in our class drive and share, because I wanted to show you where we have some data. I showed this in the other video, but now we're going to pull some covariates out so I can do some analyses. So in the resting state fMRI dataset folder, this is where we have the input data, everything that we imported, and it also has some process data. So if I went into each of these directories, we're going to see that there are a lot of files created. Now you can look online and see what these different file names mean. Some of these are going to be different type of data that you could use in other software. But from here, this participants TSV file, that's one that I want to open in Excel so we can import in. So I will highlight that and click download, and then that has now downloaded. So let me open that, and I'll open that in Excel. And that'll just take a second. Okay, so here it is in Excel. And in this file, we have participant ID, age, and gender. So I'm going to do 2 things. I am going to copy the age as a covariate into there, and then I'm going to do something with gender so we can do several analyses. So in here I had recoded this. Initially it said male and female. I just recoded it as 1s and 2s and randomly picked one to start. So we ended up male being 1 and female being 2. But really we can do 2 things with this. So if we want this to be a covariate in analysis, we can leave it as it is. If we want to use this as a grouping variable, meaning if we want to be able to really directly compare male and female, it's best if we set it up like this. So I'm going to use some code. If that cell equals 1, I'm going to code that as a, here we go. If that cell equals 1, I'm going to code that as a 1. If not, I'm going to code it as a 0. And so I can click and drag that down. That's a simple if logical test. So if the contents of this cell equals 1, put a 1. If not, put a 0. And now what that does is that has every male participant has a 1 and every female has a 0. And so I'm going to call that one males. And then females. Over here, we just do the opposite. So if this equals 2, I'm going to make that a 1, otherwise make it a 0. So now we have two variables, and one is a 1 for male and a 0 for female. One is a 0 for male, 1 for female. And that will allow us to do a direct comparison between. And now we can do two more things. What if we want to only look at our male participants or only look at our female participants? So oftentimes I do this, again, so we can look within a group. This would be too small of a sample really to do too much with this, but just to show you this. So then I create two new variables, and we'll do this. So I can just take this one again, if, or either one, doesn't matter. If this equals a 1, I'm going to code this as a 1, else I'm going to code it as something called nan, not a number. And so within CONN, that will read that as a missing value. And then I'll do this the other way. So now if female, you get a 1. If not, we get a not a number value. And click and drag that down. Well, it seems complex, and some of it doesn't necessarily have to be done, but that's a way I found to structure variables, these covariates, that are easy to understand and allow me to look just within a group. But for now, let's just take our age values. So I can copy these, and I'm going to come over here, and I'm going to open up a recent project. In this case, I have multiple ones, but the one that was for this class assignment was there on top. It was the latest one I'd opened in CONN on HyperGator. So that will open, depending on the size of your sample, it could take a while to open. But this was pretty quick because it's only 42 subjects with one session, a single time point. But now we need some covariates. And you can do this when you're setting up your analysis, but you can also do it after the fact. So here we go, covariate second level. And now we've added other ones. So you can add these in. So number of valid scans, invalid scans. And what this shows is there is this one person, subject number 6, who had what was called invalid scans, meaning that they had too much motion on those relative to the kind of the baseline. For the most part, people were pretty low. This is a clean data set. There are a few who had more motion, and what happens is those get motion corrected, or they can even get removed from the analysis. In some cases, these are outliers, and which can be okay. But what happens is, you think about it, this is 60 volumes. So we look at the total number of valid scans. And we need to look, some of these people have more valid scans. So let's figure out why. Why do those people have more? Could be that their resting state sequence was longer. That is kind of odd down there, but the rest is fine. I think some of the participants, looks like they might have got a little bit longer of a scan. Something's going on. That's quality assurance. Again, for our homework, don't worry about it. We're not going to get into there. That's a little bit more advanced. Again, if you're going to do this for a research project, you need to understand these quality control measures, read through the tutorial, spend time looking at the questions online and discussion online about how to deal with all of this. But I wanted to show you, these are now covariates that have been created that you could potentially add in to your analyses. So now we're going to correct for maximum motion, that there are some people here who moved their head more than a millimeter. Here are a couple people who moved more than 2 millimeters during the scan session. And that's a big enough motion. If you remember on the processing, we were wanting to highlight any motion that was more than a half of a millimeter. And we had some people who the maximum displacement that they moved their head was 2 millimeters during a scan, which is fairly large. And we could say, well, now we just want to exclude those people altogether. Again, the approach to this depends on what's been done. And that's not what we're going to cover in this class. But we're now going to add this other covariate. So I can come here, and so there's covariate tools, but I can just click at the bottom, and I'm going to add a covariate name, age. So the values, they're here in a row, but we can paste right in. So how does that work? If I highlight those and delete that, now remember we're on Hypergator, so we have to use this clipboard. But what I can do is I had already copied those values from Excel, and then I can paste them into the clipboard. And then from there, I can paste them in. So I just did a middle mouse button click, and that pasted in. You might be able to do a Control V or even a Command V to paste in, but that worked for me. And we can see the ages. Now there's not a huge age range because these are all college age individuals, sort of undergrad, graduate school, very likely who's part of this sample. And so this is a young sample, and so if we were going to do an analysis to look at associations between age and connectivity, this signal from the resting state probably won't see very much. So that is age. Now we do have some additional covariates we can add. You don't have to do any of this. For screenshots, we can just look at intrinsic connectivity. But I'm going to add in these 4 other covariates. So this will be males. Oops, that somehow went on the other screen. Males, here we go. I can highlight these and delete those. When I come back to my clipboard, I clear it, I paste in those values, delete the extra row at the end, do a middle mouse button to click it, and then I can do the same thing. Females, and I will go over to this window, and we'll just go back and forth like this. Oops. Notice when I highlighted all of that, it filled it in on the clipboard, so I need to paste these back in. And then I'm going to do the males only. I don't have to do the underscore. That's just kind of a convention that I have from spending time in Linux. So we do a males only. There we go. Now this should work. And then I will do one more, females only. And you can have a space in the file names. That's okay. This is really just a variable name, not even a file. File names, we don't really want spaces in Linux, but here it doesn't matter. get rid of the extra one, middle click, and now we're done. So at this point, I like to save it. This will just save the project, and if something happened, HyperGator exploded, then in that case where everything's gone, so it doesn't matter. But if my computer crashed, HyperGator is still running, or the job ended, realize kind of where I was getting at. If somehow I didn't give enough time, I'm running out of time, and HyperGator would end this process. Again, this is a virtual computer that's being run. HyperGator could just run it if it runs out of, or it would end the job for run out of time, and then that's it. And if you haven't saved your work, then it's lost. So I save, and each step tends to save automatically, but in something like that, we need to do this saving. Okay, now we are going to look at the results. So over second level, we have two options. And for homework, you can do what's called either a seed-based analysis or an ROI-to-ROI. So we have, the seed to voxel, they're both seed-based, but one, we will do an analysis from an area of the brain to everywhere else in the brain. The other one will do connections between set regions of the brain, and I'll show both of them. So here's the ROI to ROI. And so we want to include all subjects. Let me just show you all subjects. This will take just a little bit of time to compute the results. And all this is asking, as it says right here, does the average connectivity differ from 0? Meaning, are we going to be able to see these connections between different regions of the brain? So that has finished processing. This is everybody together, all 42 participants. We're looking at connections. Now, there are a lot of connections here, and really we're showing more connections than we need to. So there's 164 regions of interest, total of more than 13,000 connections. And what we really want to do is select a subset of those. And so one way to do that is to come and click on this. It's a little bit slow sometimes. And I want to use only these, for now, let's just use these networks. And I'll click on that and I'll tell you what those are. So CONN includes two different atlases of the brain. One is a structural atlas. So there is essentially a little sphere that's placed in different parts of the brain. This is what we would call the superior temporal gyrus, or this is the posterior cingulate cortex. Just different structures within the brain. And if you know neuroanatomy, then you can know where those are. But it also includes an atlas that are functional networks. And so these are based off of either sort of group level resting state networks, so just kind of what we call kind of intrinsic connections in the brain, or they've also pulled some of this from task-based analyses. So, and these are averages based off of previous research. But what we end up with are here's a language network, what we call the salience, sensorimotor. I know this is a little blurry. Dorsal attention, a visual default mode, cerebellar, and a frontal parietal. And the naming of those is slightly different from group to group, but default mode and salience tend to be called the same, and some of the others visual. You can use other atlases. This has built-in atlases. There are other ways to look at the brain and parcellate it, as it's called. And there's no one best approach. This is just what's the default. If you want to use different atlases, you can do that. There are ways to add those in and do the processing. And we will not cover that. But this will show connections using this graph network. Anything that's going to be blue is a negative correlation So that's what we call an anti-correlation. Anything in orange or red is going to be a positive correlation. So if we look within networks, we tend to have strong connections. And that's how they get to be a network. We have these strong connections within network. Some of these networks we have at least moderate positive associations between networks, but some of them we have strong negative ones, like salience to default mode. This strong blue, or the sensorimotor to frontal parietal. So those are strong anti-correlation, and that can be important. What happens sometimes with broadly what we call brain damage, Alzheimer's disease, or traumatic brain injury, or as I talked about in class, after major surgery under general anesthesia, we can see weakening of these anti-correlations, meaning that these networks are not quite as strongly, there's not that strong negative correlation between them. We can sometimes get the correlation closer to zero, which is not a sort of the ideal state. Meaning that when default mode is active, and it tends to be active when you're not doing much, then the salience network is not active. When the salience network is active, default mode is suppressed. And that's because we think salience is helping to turn the default mode off and then turn on these more frontal, central executive type of planning and processing networks. So we can see positive and negative correlations can come over here. So you could submit a screenshot of that, and that would be fine. This is just inherent to everybody. This is what connectivity looks like. Again, we haven't really done any interesting of analysis. This is just all 42 people. Do we see these brain networks? And but then we can get into age, or is there a sex or gender difference, or whatever covariates you have. But if you click on that 3D brain, I'll make that a little larger, then we can get a sense of where these are. I don't know if I should have made it quite that large. It tends to be a little bit slower trying to do this visual kind of 3D view on here. But you can click and drag and rotate this, and that'll be slow because it's a large image. And again, trying to do HyperGator, or the way I'm displaying HyperGator, not strong graphics capabilities. So you can get different views of the brain, and you can add different surfaces. You can make it look more like a brain. This is just sort of the space inside the skull, essentially. But you can use these for a screenshot. So I can do a screenshot, and we can look at this inherent. These are sort of general areas of the brain, strong connections, and strong connections in positive direction and negative direction. These are connections there. So those are the resting state, or these functional networks, I guess I should say. We could also do this from the atlas regions of interest. Again, this is more of a structural understanding of the brain. And we're going to see how the different parts of the brain are functionally connected. Is there a correlation in bold signal within these regions, between these regions? So this will take just some seconds to load. There's more regions here, about 132, as we can see. So 8,600 connections, and a lot of these are cerebellum. But we can see, again, there tends to be this bias toward positive connections in the brain. There are certainly some negative connections, a lot of blue, but we tend to get more of the positive, and that could reflect reality. It could be a potential issue with functional imaging in general, but we think it reflects reality. Again, we can do the brain. We could also click on this top one. So before I'd clicked on the bottom one to select, and I could select a subset. So let's say I had a specific hypothesis that I thought that as people get older, for example, we're going to have weakening connections between part of the frontal lobe. So this middle frontal gyrus, that includes what we call the dorsolateral prefrontal cortex, which is important for working memory, planning, organization, all sorts of tasks. Maybe we'll say there's going to be weakening of connections between that and part of the parietal lobe. So we could make very specific hypotheses and say, I'm only going to look at these 4 regions or these 2 regions, just connection between there. And so we don't have to look everywhere. Again, we can do hypothesis-driven research. And this should simplify considerably. And what we see is there are positive correlations between left and right of each of these structures, and then generally positive correlations between them. Now, I said, what about the age piece? We're not looking at age, we're just looking at inherent connectivity. If we want to see where those are, roughly have to know anatomy, but this is sort of the top part, close to the top part of the frontal lobe, close to the top part of the parietal lobe. And so these are functionally connected regions. So again, any of this, you could do a screenshot of these, submit those as homework. Now, if you wanted to do maybe a little bit more interesting of an analysis, I'm going to just close that window, don't close CONN, but now I'm going to highlight age. And let me just highlight males. So this is where typically I will also do another variable that would be sex or gender, whatever we're going to call this depending on how it's defined, but we can use that as a control variable. In this case, it's okay because, again, there's just 2 levels, males, females, they have a different value, and we can control for this. So I'm going to control for sex, and we're going to look at the effects of age. So how do I do that? I can click here and do effect of age. So notice we have a 0 for all subjects, and what that just means is that's really not our variable of interest. And then I have a 1 for age and a 0 for males. So that means we're going to look across everybody, and we're going to covary for the effects of sort of everybody. That's a little bit complex statement. We're going to covary for the effects of sex, and in this case, males, but again, it's 2 levels, 0 for female, 1 for males, and so we can just use that information to say there's this person, there's a different, these people, and meaning some people have a 0, some people have a 1, and then we can covary for that. So this will covary for sex, and we're looking at the effective age. So I can click on compute results again. This should be pretty quick. There's only 42 people, not a very complex analysis. It is complex, but at this point the computer has done all the heavy lifting, and this is one of the good things about CONN is it's pretty straightforward to use. But the downside of that is it's easy to point and click and do some analysis. And if it's not theoretically grounded or you don't have a pre-existing hypothesis, then you're just pointing, clicking, exploring, and that gets into, you're just kind of on a fishing expedition, and that's not necessarily good science. And so what I recommend is not doing that. We want to have specific analyses that we're going to do. Okay, so what are we looking at here? Nothing. Why is there nothing? Well, it's because so far there is no effective age. So let's see if that's true with a smaller subset. So this was mixing both the structural and functional networks, which I do not recommend doing. I do just because there's going to be overlap, so you want to pick one or the other or do subset. And here, we're not seeing any results. Now, we are looking at 32 regions of interest, almost 500 different connections, meaning there's essentially 500 different analyses that are done. So we correct for multiple comparisons. Maybe you don't need to correct for multiple comparisons, or maybe you need to change this. This gets into statistical background. So if you don't know what you're doing, again, you need to do some background reading about how you're going to change this. But typically, any of these first three are going to be what we consider robust statistically. But the caveat is, here we're saying that we're expecting to see an age effect across all of these connections. And maybe that's not true. Maybe there's just going to be specific subsets. In that case, there are different ways to adjust for that. Say, instead of looking across this whole network, maybe we're going to look at everyone in the network, but we only want to allow for specific connections to be significant, even if the whole network doesn't show differences. But just to show you, to see if there's even a hint here, I'm going to not correct anything. So we'll have a P of 0.05 and then no cluster threshold. So this is not robust statistically. We have a relatively limited sample size, but again, this is not what I recommend for a real research project, just showing this to you. However, I'm showing it to you because we can see some potential patterns here. Now, why am I doing this? Remember, we are looking at the effects of age. We have a relatively small sample size. It's not terrible, 42, but we have a very limited age range. So these are pretty much everybody in their it's in their 20s. I don't know if we had anyone outside of that range, but for the most part, right around kind of 22, 23, 24. So there's just a couple years difference there. And so we're not really getting any signal. If we had 20-year-olds all the way up through 80-year-olds, then we would very likely see some differences here. See some association, because we have more range that can co-vary with the connectivity values. But one thing we can get a sense of, and this is all between, so red is a positive association with age, blue is a negative association with age, and we're getting a hint, again, this is completely not statistically robust, but we can kind of get this hint that there might be some changes and some, I don't say weakening, these are actually potential. Blue, we could end up, this is where it can be hard to interpret, but blue is a negative correlation, but that doesn't mean a problem. That this can actually be, if we notice, these are between networks, and we can have a strengthening of this negative correlation between networks, so they can, in effect, with more age, or maybe becoming more anti-correlated. They could be getting weaker. You have to look at the effect size. We won't go into that, that's more advanced, but interpreting direction of this, what these mean, takes some time. You have to look at values, and there are ways to do that. I can click here and we can get a sense of what's going on. Maybe we'll see if this works. I don't know if that, let's try just one of these. This particular single connection right there. I don't know if that's going to show anything. Anyway, so we'll ignore that. We don't have enough signal for this to really show, but you can look at some of these graphs. There's things you can click around on. But again, we can look at some patterns there. So we didn't see an effective age. There just isn't enough range in age, really, to see anything. What if we wanted to see, is there effect of, I'm going to actually take age out. Is there a difference between here? So do we have areas where females have greater connectivity than males. This will be a simple kind of sex difference. And the caveat is higher connectivity could mean two things. It could mean more positive, which could be good. It could also mean less negative, which isn't necessarily good. So why does less negative? Again, that's more positive. You're closer to the positive direction. But if we have two areas of the brain that generally we expect them to have a strong anti-correlation, so they should have its negative correlation, then if you are closer to zero or positive, so let's say we have two areas of the brain, like a salience network, default mode network. Those aren't areas, but these networks, they should have, let's say they have on average, a negative 0.4 correlation. You could have a positive association, or it's sort of this positive, if you have a group difference and we're testing one group has higher connectivity than the other group, well, the higher connectivity means that one group might be, instead of a negative 0.4, they're a negative 0.2. And that's not necessarily good. So again, we have to be cautious in interpreting. Let's limit this just to these network regions of interest. So this will only be these 32 regions of interest. We're not seeing any differences in a relatively small sample. I think we have 42 overall, but we have maybe 16 to 18 male participants. But in general, we're really not seeing any differences. If I do a kind of a very, very weak, again, not robust statistical analysis, we can see generally here we're seeing more red than blue. So there's sort of this hint that female participants, so what did we test? If females have higher connectivity than males, and so that would be this red. And so kind of generally, that's more the pattern we're seeing. Blue would be where males have higher than females. But again, not robust statistically, so this is definitely not something we could publish. And then let me just show you, let's look at the structural regions of interest. Let's see if we see anything. Again, for research, generally you're going to have some hypothesis. There's a lot there. There's a lot of regions of the brain, and I'm not correcting for multiple comparisons. So let's go back to that. And there's different ways to do it. Again, you have to understand the statistics behind these corrections for multiple comparisons. When I do a kind of a full correction for multiple comparisons, we don't see anything. But again, we have small samples. We have 42 total, and as I said, only about 16 to 18 males. A few more females than that. But that means now we're only testing about 16 to 18 versus 24 or so, and that's not a lot of people in each group. So we are underpowered for the analyses. And that's where we might be a little bit more or less stringent with some of our statistical analyses. Again, that's a different question. So any of this you can run. You can submit screenshots of the connectivity, sort of this circle. You could do the 3D brain, do screenshots. Yeah, I would just recommend just kind of do a screenshot on your computer. You can export and save the images. Let's try one more thing. Let's do this. I won't correct our clusters. Our connections will only look at ones that are less than a p-value of less than 0.01. We'll see what we If we see anything, we should see something. Let's maybe make this 0.001, which at this point is getting to be relatively robust. And meaning that given our small sample size, which isn't great for a lot of reasons, but this might be area, we don't want to have too many false negatives. We don't want false positives, but again, there's going to be a balance, and this can be one way to potentially balance that. If you had more of an exploratory sample, this preliminary data or pilot data, we could kind of show there might be stronger functional connectivity in females than males. Whether or not that's good or bad, we don't know. Really probably can't even label it as good or bad. But again, if these were clinical groups, we had individuals who had traumatic brain injury versus not, and they're matched in all of these other variables. Are there differences? And if there are differences in connectivity, is it good or bad? It depends. And just because there's a group difference doesn't mean we can always interpret one as superior, inferior, or better or worse. We might be able to, but again, something to keep in mind. But potential differences, so you could screenshot this, do a 3D brain, screenshot that. You can just do the simple all subjects analysis right here without having to do anything other with the covariates. Submit a screenshot, meaning just display your results. If you've already calculated those results, you can just load the results, and it should be pretty quick to load. So you can load it, take a screenshot of what we see. We'll have a lot of connectivity here showing up because we're looking at 13,000 connections, and this is just intrinsic connections. And that will be it. I would make that a little bit larger a screen if possible. Take a screenshot or maybe do a subset of connections, and we'll call it good, and that'll be it for the assignment.